healthy age-matched neonates, and this is an ongoing project currently in the lab, so maybe we'll talk about it next time. So for today, I'm going to focus mostly on the T-cell memory part. So all of you know that whenever there is a disease, it is going to develop an immune memory that leads to the natural immunity. And this is what exactly has been exploited for the vaccination, where through vaccination, we induce immune memory and eventually the natural immunity, and that eventually controls the active disease. Okay. So for people who are not really T-cell biologists, I just want to introduce you a little bit to what T-cells. T-cells recognize the pathogens through their T-cell receptors, which are present, the antigens that are presented by the antigen-presenting cells through MHC molecules. Upon this recognition, there is a second signal that comes up where there is the co-stimulatory molecules get activated, and the third signal comes from that of the cytokines. The T cells have a dichotomy. There are majorly two types. One is the killer cell, another is the helper T cell. And helper T cells provide help to various other immune cells, including that of the CD8 cells, which get activated and make these cytolytic molecules and kill. I don't have to introduce this aspect because you already heard from multiple speakers today and yesterday that there are cytotoxic CD4 cells, and these are relatively less well studied, whereas the CD4 cells are better understood for their helper function as classified based on their cytokine melee and the transcription factors they express. So the T-cell response is a multi-step process where the T-cells rapidly expand, which coincides with that of the increase in the viral load or any pathogen. And upon the resolution of the infections, the T cell contract and leaving behind a very small portion of the cells as memory. And that's exactly what we are going to talk about today. So while some individuals are maintaining this memory better, the other individuals fail to do so, and they actually form the protective and the susceptible immune response respectively. So upon a secondary infection, if you have maintained the better immune memory, you're going to mount a better immune response and control the infection subsequently. So now if we want to say what is a good memory, so the memory has to be long lasting. Once you have an infection, it should really be there for a very, very, very long time. And it should have a potential to differentiate into effector cells so that it can control the infection whenever there is a secondary infection. And should be able to move to the site of infection, that's the mobility, and be able to self-renew even in the absence of antigen. Okay? So in CD4 cells, while the other classification of T helper cell, which is Th1, 2, and 17, has been better understood, the classification based on the developmental stages of the memory has not been that well understood. But out of that, there are markers which distinguish them into different developmental stages, like stem cell-like memory, central memory, effector memory, and then effector memory expressing CD45RA. We and others have shown that this TEMRA compartment is indeed enriched for the cytolytic molecules and forms a bona fide CD4-CTL compartment. So, sorry. So in my previous part where we did using single cell, we showed that though this was thought to be terminally differentiated, it actually was heterogeneous, and we found a long-term kind of memory within this TEMRA compartment. And further, using the T-cell receptor sequencing, we have shown that this actually is the precursor to that of the CD4-CTLs. So if you visualize memory, what is the developmental lineage? Is it a very linear developmental stages, or at every stage there is a bifurcation that leads into a different kind of memory? And what are the specificities to different infections? But in reality, what exactly exists is this kind where you have heterogeneity within each of these compartments. And this heterogeneity could be as subgroups within the memory that can indicate a developmental lineage, or it could also come from the specificity. So for specificity, we have to look into antigen-specific cells, and the characterization of antigen-specific memory T cells is relatively difficult because of their rarity. Okay? So you imagine that you're going to get 10 to 100 cells out of 1 million PBMCs. So it's a very, very rare population. So for that, what we did to be able to understand the specificity, we took the PBMCs from the donors who were seropositive for various infections like CMV, dengue, and then our influenza or COVID. And then also tested them for their responsive to the peptide pool stimulation, and sorted the activated T cells based on the surface expression of co-stimulatory molecules such as CD137 and 154, and sorted these activated cells across different CD4 memory compartments and pooled them in a ratio so that we have a representation from each of these memory compartments. To be able to compare the CD4 CTLs to that of CD8 CTLs in the same environment, we also sorted cells from the CD8 compartment, which was the effector memory. Then we performed single-cell RNA-seq, and we multiplexed using the H2Os for each donor, and we also added some of the memory markers at the oligotagged antibodies to be able to distinguish them in the single-cell data, and then we sequenced them. So for now, I'm going to mostly focus on talking about the CMV, and at the end I'll show how this is in relative to the other viral infections, what is the characteristics. The CMV, we found about 9 clusters, and they were distributed across the different memory compartments. We had the effector memory and as well as the long-term memory, and they were also aligned with the antibodies that we used, oligotagged antibodies that we used in while doing the experiment. So this suggests that we had a representation from different memory compartments, and then we looked into their functional characteristics in an unbiased way. Sorry, it's actually moving very fast. We had several clusters which had a signatures of long-term memory, and also we had a cluster which had the cycling cells, very small portion of the cells as cycling cells. And we had, interestingly, a cluster which had interferon response signature, and a cluster that was showing T regulatory signatures, and the cytotoxic, that is the CTL clusters. So, very interestingly, the Treg cluster showed the expression or the coincided with that of the interferon response signature. So if we look further into that cluster, so you see that the co-expression of the Treg specific markers show that this is indeed a Treg cluster. And they actually had an entire signature of Tregs enriched in those clusters, and that had an overlap with the interferon response signature. So the next question is, are these Tregs suppressive in nature? And many of the molecules that are linked to their suppressive function were actually enriched in this cluster. But however, they did not really make suppressive cytokines such as TGF-beta and IL-10. We have many other evidences. In the interest of time, I'm going to skip that. We also saw that these clusters express GATA3, not the TBET21 or the ROR-gamma-C, indicating that they're potentially Th2 suppressive Tregs. And if anyone wants to hear more about it, I'll be happy to talk later. So the next one we focused was the cytotoxic clusters, where we had 2 cytotoxic clusters. The question is, what is the difference between these 2 cytotoxic clusters? And that actually indeed coincided, the CTL signatures coincided with that of the TEMRA signatures. And you can see that the cytotoxic clusters express granzyme, porphyrin, which are known for their CTL function. And many of the molecules were commonly expressed, for example, porphyrin and NKG7, whereas these 2 clusters show variability between many of the transcripts that we observed. One was granzyme. The granzyme expressing cluster also made the cytokines, while the other cluster expressed a higher amount of granulysin compared to that of granzyme, and they did not really make the cytokines. And the late effector specific transcription factor, like ZEP2, was expressed in the cluster that was making cytokines. The other cluster was expressing a slightly earlier transcription factor like EOMS. So indicating that they're probably at a different developmental stages. We call them pre-effectors and effectors. Sorry. Yeah. And not just these, sorry, I don't know why. Yeah, not just this, the interferon gamma and TNF cytokines, they also made many chemokines, which were highly expressed in case of effector cluster compared to that of the pre-effector cluster. Next, to be able to say that these are effector and pre-effector, we looked at the T cell receptor repertoire. Both of these clusters were clonally expanded, and you can see that some of the clones were found in as high as 144 cells. So it's a very highly clonally expanded population. Now, if I take only the top 25 most expanded clonotypes, we found that this proportion of the cells expressing these top 25 clonotypes were more in cluster 2, which is the effector cluster, compared to that of the pre-effector cluster. So the next evidence we wanted to ask was, are they developmentally linked? So we did a pseudo-time analysis with the clusters that are somehow associated with the CTL cluster. One was we used the cytotoxicity signature to take those cells, and the sharing, clonotype sharing between two different clusters that shared clonotypes with that of effectors, that the cytotoxic clusters with the any other cluster. And based on that, we used cluster 0, cluster 3, cluster 7, and 2 and 4, which were the cytotoxic cluster. As you can see that the effector and pre-effector clusters really bifurcate, so indicating that they're probably at two different developmental stages. Yeah, so the next question is this difference that we are seeing in the pre-effector and effector because of the CD4 and the CD8 proportion? And both these two clusters expressed both of CD4 and CD8 transcripts, indicating that they're actually clustering together. Now, is the proportion of the 4 and 8 different? That is why we are seeing this variability. So we looked at the proportion of CD4 and CD8, and those who are thinking that there are a lot of double negative cells, these are sorted CD4 and CD8 cells. We could not assign them to 4 or 8 based on the transcript expression of CD4 or CD8. So we are calling them as NA, not assigned. And we saw that the proportion did not really drastically differ between these two clusters. And we compared these cluster 2 and cluster 4, that is the effector and pre-effector cluster, and divided these cluster based on the CD4 origin and CD8 origin. And the transcriptome profile within clusters 2 for 4 and 8 was similar, the same way for the cluster 4, suggesting that the difference that we observed was not really because of 4 or 8 proportion, but it is the different stages of the development or their effector molecules that dictated their two clusters. Okay. To summarize, from CMV antigen-specific cell, we found that the cells were at different developmental stages. A lot of people use for the antigen isolation cytokine as a marker. Maybe that's not the right way of looking at it. And the suppressive Tregs were suppressive in function. They also had signatures of interferon response. And the CTLs could be classified into pre-effector and effectors based on their effector molecules that they expressed. And these different types of CTL clusters were not really based on, the distinction was not based on the CD4 or CD8 proportion. So the next question is this a characteristic feature of CMV alone, or it is also found in the various other viral infections? So we looked at two other viral infections, that is dengue and then flu. And in dengue, again, we found the clusters between late effectors as well as the early long-term memory. And the signatures again showed that there are multiple cytotoxic clusters which could be distinguished based on their cytokine secretion or the molecules that are linked to their longevity. And we also had a Tregs which were enriched for the interferon response signature. And the CTL clusters were not distinguished based on the CD4 or CD8. The same is true for an influenza virus, where we looked at there were again multiple cytotoxic clusters, and the Tregs were enriched for the interferon response. The CD4 and CD8 did not distinguish these CTL clusters. So overall, the antigen-specific viral antigen-specific Tregs are indeed suppressive in nature, and they are showing an enrichment for interferon response. And we have a lot of evidence suggesting that they could be Th2 suppressive in nature. And the CTLs were clonally expanded and came in multiple flavors. And this flavor could only be distinguished based on their effector molecules and not based on their CD4 or CD8 origin. With this, I would like to thank people who have been very instrumental in completion of this project. The project was led by my first graduate student in the lab, Ronakkar, with the help of other students in the lab, Kirti, Shreya, and Somdev. We have collaborator Anish, who helped us initially to set up all the pipelines. And Dr. Anjali Sharma is from where we get our clinical samples. And the volunteer blood donors, without who's volunteering to donate, we wouldn't have been able to do any of this. And the funding from Welcome DBT India Alliance has been of a great help, where it's a very flexible funding. They allowed us to do whatever we wanted to do. And the sepsis project is funded by Department of Biotechnology, Government of India, and NII for both financial as well as the facility support. With this, I would like to thank all of you for listening, and we'll be happy to take any questions.